Good day, everyone. Today I'm going to be speaking about the work we've done to optimize our sample preparation method for the detection and quantification of allergenic proteins by LC-MS. Just a quick introduction. My name is Khyla. I am a research assistant at FACTS in Stellenbosch. I am involved in method development, validations, and optimizations. My goal is to contribute to the understanding of allergen testing. One of our biggest challenges we encounter during sample preparation stems from the sheer diversity of food matrices from which allergenic proteins needs to be extracted. The term food matrix has been described as a physical domain that contains and/or interacts with specific constituents of a food, providing functionalities and behaviors which are different from those exhibited by the components in isolation or free state. When food has been heat processed to some degree, protein aggregation or other chemical reactions such as the Maillard reaction can take place. This renders proteins less soluble, and if your sample preparation is not optimized to overcome this may result in quantification errors or even false negatives. The objective of this study was to optimize the sample preparation procedure, which aims to solubilize proteins from a food sample. This is the first step in our workflow and takes place before reduction, alkylation, and digestion. The main target of the extraction procedure is to obtain the best protein quality and yield. Selecting an appropriate extraction method has to be the first step before further analysis. The success of the LC-MS proteomics analytical method lies in a sample preparation procedure that allows efficient detection and quantification of allergens in the subsequent mass spectrometric analysis. The ideal sample preparation protocol would be a method allowing the total extraction of all protein components from a proteome of the sample, whilst keeping contamination from other molecules to a minimum, and lastly, enabling a high level of reproducibility across all types of food matrices. The factors we considered in our quest to optimize our extraction procedure was the extraction buffer, the pH, the temperature, and the homogenization method. We compared two sample extraction buffers across a wide variety of sample matrices, one containing 1% STS and the other a PBS buffer commonly used in ELISA test kits. The efficiency of each buffer was based on the total protein concentration as determined by Bradford's assay. On average, we saw a 25% increase in protein concentration from the STS buffer compared to the PBS buffer. So far, we have shown that the STS buffer is able to extract more total protein than the PBS buffer. To see whether this tendency holds up in the extraction of target proteins, allergen-spiked samples were analyzed by the LC-MS. 3 variations in sample matrices were spiked with a 1000 ppm egg, a 1000 ppm almond, and a 1000 ppm peanut. The vegan cookie represents a carbohydrate-rich matrix. The low-fat cottage cheese represents a high-protein matrix, whilst the temperate chocolate represents a fatty matrix. We were unsuccessful in obtaining protein from the spiked cookie and thus excluded this matrix from the experiment. We suspect that the flaxseed ingredient might have caused protein aggregation to take place, a phenomenon we have seen with the closely related fenugreek seed. We plan on exploring this further. From the STS page gel, it is clear that the STS buffer extracted more protein than the PBS buffer, with some bands only appearing for the STS buffer. We compared the peak areas of our selected almond, egg, and peanut peptides on the LC-MS. The STS buffer resulted in a 38% higher peak area compared to the PBS buffer. Thus, the tendency did hold up. The STS buffer is able to extract more target proteins. The success of the STS buffer is likely related to the STS. The anionic detergent's ability to bind proteins through ionic and hydrophobic interactions leads to the denaturation of the tertiary protein structure, increasing its solubility. This leads to optimal extraction from food matrices. After determining that the STS buffer is the optimal buffer, different allergen standards were extracted at different pH levels, using the STS buffer at a pH of 7, 8, and 9. No clear differences were observed on the STS page gels. The protein concentrations of those same extracts were determined via Bradford's assay. There were no significant difference between the protein content at the different pH levels, with a relative standard deviation of only 4%. Results indicate that the addition of the surfactant, STS, plays a larger role in the extraction of proteins, rendering factors such as pH less significant. To determine the best temperature for extraction, 3 different samples were extracted at 4 different temperatures. The STS buffer was heated to 40, 60, and 80 degrees Celsius, or left at room temperature. In the peanut butter sample, bands appeared at 40 degrees Celsius that was not visible at room temperature. At 60 and 80 degrees Celsius, the protein yield was clearly much higher. There were no significant difference between 60 and 80 degrees Celsius. The oat sample also showed an increase in protein yield at 60 and 80 degrees Celsius. A mixed sample consisting of equal parts oats, peanut butter, soy flour, salted butter, and beef patty were included to represent a complex food matrix. One protein band was not visible at room temperature, appeared at 40 degrees Celsius. Protein yield was slightly improved at 60 and 80 degrees Celsius, and there were no significant difference between 60 and 80 degrees Celsius. Samples were homogenized in the STS buffer by reducing the particle size using a sonicating water bath. By homogenizing the samples properly, we can ensure that the thorough protein extraction can take place. The ultrasound of the sonicator causes proteins to undergo a physical disruption. It heats by specific absorption of acoustic energy, dynamic agitation, and shear stresses. The mixed sample was used to test the optimal sonication time. At 1 hour, these bands showed a higher protein yield. The sonicating water bath did present some challenges. Due to our large testing volumes, the sonicator was oftentimes packed. This may prevent ultrasound waves from reaching all the samples, causing some to be inefficiently homogenized. The sonicator also adds an extra hour to the extraction procedure in order to extract the most proteins. We started exploring alternative homogenization methods, one of which was to vortex the sample in STS buffer with the addition of a few 3 mm glass beads, followed by a 15-minute step in a shaking water bath heated to 60 degrees Celsius. Samples were extracted via the different homogenization methods and analyzed on the LC-MS. The results look promising for the spiked temper chocolate. There was a 20% increase in the peak areas of the peanut quantifier peptides when homogenizing with the beads and water bath. A spice matrix containing 70 ppm milk showed similar results. The water bath and bead method managed to increase the peak areas of the milk target peptides by 12%. Similarly, the cookie matrix showed a 13% increase in peak area for almond and milk target peptides after homogenizing with the water bath bead method. When we consider the overall extraction results, it can be concluded that the STS buffer allows for a higher protein yield and that the surfactant, STS, plays a big role in the solubilization of protein, rendering pH a less significant factor. The extraction temperature indicated a higher protein yield at 60 degrees Celsius or more. And we also determined that the homogenization method has an important effect on the total amount of protein detected on the LCMS. It was also found that a combination of the shaking water bath and glass beads yielded a level of target proteins that is similar or higher to that yielded by an hour in the sonicating water bath. Thank you.